Hey everyone, and welcome. So if you've ever stared at a single-cell dataset, you know that feeling, right? It's just this massive universe of information, and it can feel like pure, beautiful chaos. Today, we're going to demystify one of the most important steps in making sense of it all, how the Seurat toolkit uses this really smart graph-based clustering to help your cells find their true biological cliques. Let's dive in.
Okay, so let's just picture the challenge we're up against. You've got data from thousands, maybe even millions of cells. And for each one of those cells, you've got measurements for thousands and thousands of genes. It's kind of like being dropped into the middle of a gigantic, bustling city and being told, okay, go map out all the different social circles. I mean, where would you even begin?
And this is what it looks like in practice. A giant, unstructured cloud of dots. Every single one of those dots is a cell, but right now, they're just one big, undifferentiated crowd. Our mission, should we choose to accept it, is to find the hidden structure, the hidden order inside this cloud. You know, the cliques, the communities, the potential cell types just waiting for us to find them.
So why is this so mind-bendingly difficult? Well, it all comes down to something called the curse of dimensionality. Sticking with our city analogy, it's like trying to group people based on 20,000 different personality traits all at the same time. I mean, it sounds super thorough, but in reality, it's just computationally overwhelming and, honestly, statistically noisy.
You see, the real kicker is that when you're dealing with that many dimensions, our normal ideas of distance and similarity kind of fall apart. Everything starts to look equally far away from everything else. It makes it almost impossible to tell which cells are genuinely similar and which ones just happen to look close by random chance. It's like everyone in the city is a distant acquaintance and nobody is a close friend.
So how in the world do we get around this? Well, the first step is to simplify. We've got to cut through all that noise and focus on what really defines each cell's identity. And this is where a super powerful technique called principal component analysis, or PCA, comes into the picture. Basically, what PCA does is it takes those thousands of gene measurements and boils them all down into just a handful of what we call principal components. You can just think of these as meta features. So instead of 20,000 different personality traits, we're now describing each cell by its most defining characteristics, like maybe being highly metabolic or primed for proliferation. Each one of these meta features is actually a combination of genes that work together, capturing the major biological stories in your data.
So we're starting here with this massive, noisy, overwhelming matrix of thousands of gene measurements for every single cell.
And we boil it all down to this. Now, each cell is just represented by its scores along a few of these powerful metafeatures. We're not in a 20,000 dimensional space anymore. We're in a much, much simpler space, maybe just 10 or 20 dimensions. And that makes it way easier to meaningfully compare cells.
All right, so we've got our simplified personality profiles for each cell. Now what? Well, now we have to actually build their social network. And that's exactly the goal of Seurat's FindNeighbors function, to figure out which cells are most similar to each other in this new, cleaner PCA space.
And Seurat has this really clever two-step process to do it. First, as you can see in step 1, it finds the K closest friends for every single cell. Those are its K nearest neighbors. But, and this is the important part, it doesn't stop there. In step 2, it refines those connections by looking at the shared friends between any two cells, and that builds a much more robust shared nearest neighbor, or SNN graph.
But hold on a second. You might be thinking, why the extra step? I mean, if two cells are close to each other in this PCA space, isn't that good enough to say they're similar? Why do we need to check if they share friends?
And the answer is just so intuitive. It's because just being close to another cell, especially in a really dense neighborhood, can be totally misleading. It could just be by chance. A much, much stronger sign of a real relationship is a shared social circle. The more friends two cells have in common, the more confident we can be that they actually belong to the same community.
And this visual just nails the concept. Take a look at the left. That's the initial K nearest neighbor graph. It's kind of messy, right? It connects any cells that are simply nearby. But now look at the graph on the right. That's the SNN graph. It gets rid of the weak, random connections, and it strengthens the connections between cells that have a lot of mutual friends. And in doing so, it starts to reveal the true, tight-knit communities that were hidden before.
Okay, so how does Seurat actually put a number on this? How does it measure that connection strength? It uses a metric called the Jaccard similarity, or Jaccard index. This score is exactly what Seurat uses to add a weight to the connections in that SNN graph, and it's a beautifully simple way to measure how strong a relationship is.
Now, don't let the math here scare you off. It is way simpler than it looks. All it's saying is this. Take the number of friends two cells have in common and then divide that by the total number of unique friends they have between them. That's it. A high score means they have really similar social circles. A low score means they don't. Simple. Okay, we are on the home stretch. We've built this amazing, robust social network. We know who's strongly connected to whom. So the final step, which is done by Seurat's FindClusters function, is to actually find the distinct cliques, the communities, within this whole network.
And this is exactly what we're trying to do. We use a community detection algorithm. Seurat often uses one called the Louvain method, and we apply it to that SNN graph we just built. The algorithm's job is really simple. Find groups of cells, or clusters, that are way more connected to each other on the inside than they are to anyone on the outside.
And here's the really cool part for you, the researcher. You are in the driver's seat here. The most important knob you can turn is the resolution parameter. If you turn it up to a higher value, you'll get more, smaller clusters. It's like zooming in on the sub-neighborhoods. If you turn it down, you'll get fewer, larger clusters. This lets you tune the whole process to match whatever biological question you're asking.
So, let's just take a step back and look at what we've done. We started with this chaotic cloud of cells, we simplified it with PCA, we built a really robust social network, and then we identified all the key communities. So, what's the big biological payoff?
And boom! This is the payoff. That initial messy cloud of cells is now this beautifully organized map. Each color represents a distinct cluster, a population of cells that the algorithm decided are super similar to each other. These are your candidate cell types, your potential cell states, all ready for you to dive in and interpret them biologically.
But here's the thing to always remember. This isn't the end of the analysis, not even close. It's the beginning of the discovery. These clusters that the algorithm found are now the foundation for your next big question. Which cluster expresses the gene you care about? How do these populations change between healthy and diseased states? The map is drawn, and now it is your turn to explore. So what are you going to find?